So nucleic acids. Nucleic acids are also biopolymers. जैसे कि we had proteins, we had carbohydrates. In the same way, nucleic acids are also class of biopolymers. So they are biomolecules and are polymers. They are polynucleotides. See the spelling TIDES, tides. They are carriers of our genetic information and production of different proteins in the body. They are of two types. One is the DNA, which is deoxyribonucleic acid. Other is the RNA, ribonucleic acid. Proteins are produced by RNA, but the message for the production of specific protein is written on DNA. So the message is primarily on DNA, but ultimately that message is taken and its final action is done. So proteins are produced by RNA finally. Okay, so this is called the central dogma of genetics, that DNA is the original carrier of information. It transfers it to RNA, and RNA then do the final activity and produces proteins. Okay, and that is how the entire genetics work. That is why it is called central dogma of genetics. Now, as we said, the word nucleotide. See, I told you that nucleic acids are polynucleotides. So what is a nucleotide? So nucleotide is having three components. One is the phosphate sugar, one is the phosphate group, other is the sugar, and third is some nitrogenous organic base. Now let us take each one individually at a time. Phosphate. Phosphate is same as you usually call sodium phosphate, magnesium phosphate. That very phosphate is to be used. That is trianion of phosphoric acid. What is phosphoric acid? H3PO4. When its trianion is formed, when we remove all the 3H plus, we have PO4, 3 minus. This is called phosphate. Okay. Now, second, what is sugar? Now, sugar is same what we have learned in case of carbohydrate, that it is polyhydroxycarbonyl. In genetic material, whether I call it DNA deoxyribonucleic acid or I called RNA ribonucleic acid, this ribo word is common. Okay. In both, I am using in both the DNA material, DNA and RNA, I am having this word ribo. This word ribo is derived or is taken from the sugar ribose. This ribose sugar is a pentose sugar. It has 5 carbon in it, pentose sugar. And day before yesterday, I taught you how to write this structure, what structure of a sugar. So see, this is ribose. Okay. This is ribose, CHO, CHOH, 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 and CH2OH. Now, yesterday I also taught you how to close the hemiacetal linkage. Right? So here, fourth oxygen is attacking on first CHO, and this first CHO is acting as anomeric carbon. If these both are on same side, then it is beta. If they were on opposite side, then it is alpha D sugar. Right? Fifth CH2H and anomeric OH. If are on the same side, then it is beta D ribose. Okay, fine. Now let us see, as all the OH are on same side. So all the OH are written on the same side. Only this CH2OH is on upside. Okay. So this is beta D ribose. When we want deoxyribose, it is this carbon number 2 of OH is removed of oxygen and is now it is deoxyribose. That is the sugar that we use in RNA. Okay. So we have learned how to draw this award for glucose. Okay. Similarly, we can have a pentose sugar. Sugar is ribose. If you just notice, all these OH should be on bottom. It is a beta. So first OH and fifth CH2OH on the same side. D. So it should be on the upside. Beta D ribose. Beta 2-deoxyribose. 2-deoxy, carbon number 2, we have removed oxygen. Okay, so phosphate we have defined, sugar we have defined. Now comes the organic nitrogenous base. Organic compounds containing nitrogen are acting as base are called organic nitrogenous base. Now in nucleic acid, heterocyclic nitrogen containing organic bases are present and are called nucleobase. They are nitrogenous base, but here they are called nucleobase. They are of two types. First is a pyrimidine base, which is monocyclic. I hope all of you sat with your register, notebook, and pen. Okay, because we need to practice the structures. So pyrimidine, it is a monocyclic base, and there are three pyrimidines. Purine is a bicyclic base. Okay, pyrimidine and purine. So see the pyrimidine and purine. But first of all, let me tell you just what is pyrimidine. Let's talk just, I'm not talking about what are the pyrimidine bases. What is pyrimidine? Okay. So it is this is pyrimidine. And what is purine? So basically this structure is replicated as such. And then we have this type of nitrogen placement. Okay. N double bond. And this is an H. First thing clear? See, end CN, this is basic structure of pyrimidine. This is the basic structure of purine. First, you should just know this. What is pyrimidine base? What is purine base? Once we are aware of what is pyrimidine base and what is purine base, then we'll talk of anything else. Okay, is that clear first? What is pyrimidine base and what is purine base? So once this structure, please write in your notebooks, reminding like pyridine, na? This is pyrimidine. This is purine. So how many nitrogens are there in purine? 4. How many in pyrimidine? 2. Okay. Now coming to the bases which are similar to these pyrimidine base which are present in nucleic acids are 3. Cytosine, thymine, uracil. I can call them CUT, cut. Okay. What will you call them? Cut. Pyrimidine, cut. Pyrimidine, cut. Cytosine, uracil, and thymine. These 3 are pyrimidine bases. Okay. This you can call PUB-AG. PUB-AG. That is purine, bicyclic, and adenine and guanine. Is that clear? Adenine and guanine. First, you just learn the names. Pyrimidine cut. You can say it PY pyrimidine. M is monocyclic. Okay, pyrimidine PYM. I can call it pyramid cut. Cytosine, uracil, thymine. PUB-AG, purine, bicyclic, adenine, and guanine. Yaad hua? Now you check the position of other things. If there are 2 carbonyls on the 2 sides of this nitrogen, then it is uracil. Same, 1 extra CH3, thymine. Cytosine is totally different. See NH2, right? There are no 2 oxygens. That is not there. Adenine and guanine are also different because there is a difference of this oxygen primarily. NH2 is there. Locations are changed, but somewhat like same. So now let us come to nucleotide we started. Now I'm coming to nucleoside side. When base is connected to sugar, now I'm not talking of phosphate, only the base, that which base, purine and pyrimidine, out of those any one base is connected to sugar. They are always connected to this one dash position. Okay, here it was OH. That OH is gone and base is connected. Let me show you back. Here it was OH, beta D ribose. That OH is gone and base is connected at this position. When base is connected to sugar at one dash position, then the nucleosides are formed. Okay, if it is connected to ribose, it is nucleoside for DNA. If it is connected to deoxyribose, then it is the nucleoside for R. I just said opposite, I guess. If it is connected to ribose, then it is the nucleoside of RNA, ribonucleic acid. If it is connected to deoxyribose, then it is the nucleoside of DNA. And also remember, out of the 5 bases, which are 5 bases, pyrimidine cut, cytosine, Uracil, Thymine. Purine ka PUBG, purine bicyclic, adenine and guanine. So out of these, the purine bicyclic are fixed. That is adenine and guanine are fixed. But these pyrimidine bases, one has T, other has in place of that U. So each nucleic acid, like either DNA or RNA, has total of 4 type of base in it. If it is ATGC, adenine, thymine, guanine, and cytosine, then it is DNA. AUGC, that is thymine is replaced by uracil, rest 3 same, then it is RNA. One of the important question is this only, which has thymine, which has uracil. Okay, so nucleoside is sugar plus one dash position per attach base. So deoxyadenosine, when it is deoxy, this is a nucleoside of DNA. 2 dash deoxy guanosine, when guanine is connected, and see the position of attachment for both adenine and guanine is from 5-member ring, this single bonded nitrogen. Okay, deoxycytidine, cytosine is used. So this again, deoxythymidine, nitrogen same. Okay, RNA has hydrogen where there is a methyl group in thymine. Just at this place, if I have hydrogen, then it will become RNA. And at these blue positions marked, all 4 will have OH for it to make RNA base. And instead of 2 dash, thymidine will then call uridine. Is that clear? So here uridine, see, same thing. OH, position of attachment is same. Just OH is extra. Adenosine, guanosine, uridine and cytidine. So basically same, adenosine, guanosine, cytidine and thymidine. Just 2 dash deoxy we have to keep adding so that you can say it is a DNA, and here you don't have to add anything, just uridine or cytidine, adenosine and guanosine. Okay, now so you have got that nucleoside is sugar plus base. When you add phosphate at 5 dash position, dash is the numbering we are using for our sugar, then it becomes nucleotide. The nucleoside bonded to phosphate linkage at 5 dash position, then it is called nucleotide. Okay, nucleotide of RNA as position number 2 is having OH and we are having uracil here. If it is nucleotide of DNA, here H is removed. Okay, and ATGC is there. DNA has ATGC, RNA has AUGC. That is the only difference. ATGC, AUGC, and the second difference is presence of this oxygen. Okay. Once such things are formed, so we have just till now talked of what is nucleoside, what is nucleotide. Now I want that all these nucleotides should connect to each other. So they are interconnected by phosphate. Okay, so we have already said that position number 5 has phosphate. Now position 5 phosphate is connected to the next molecule's 3' phosphate. Clear? Primary structure of nucleic acid is like this. The exact sequence of nucleotide in the polynucleotide is called the primary structure of the nucleic acid. Now see this sequence, this 5', this 3', this 5', then 3', then 5'. See, so one end will be where this 3' left is, OH is left open. That we'll call 3' end. And one end of this will be where the 5' end will be kept open. So that end we'll call as 3' end. Is that clear? How we name them 5' end and 3' end. Is it absolutely okay? So DNA always has two complementary strands because DNA has a double helical structure, no? So it always has a complementary strands. And remember, the 5' end is always in front of a 3' end. See, this one has a 3' end open and this has a 5'. So 5' will link to 3' and 3' down will have a link to 5'. So they are just like opposite. Okay, if this is 3' and this is 3', then see how they are placed. Opposite. Okay, 3' and 5' end are placed like this. 5', 3'. 5' is bonded to 3', 3' is bonded to 5'. And the way we had peptide linkage, the way we had glycosidic linkage, in DNA we have phosphodiester linkage because phosphate is forming esters. Phosphoric acid plus alcohol forming ester. Which ester? Phosphoester. Both sides, so diester. Okay, see, this is how it is. One yellow is a 5', then the blue will be 3'. Okay, now this is going helical structure, round, round, round spiral. But if I want to make, if I just stretch it and make it linear, then this is what it is. Now see here, you will see that different lines are present. C and G has 3 horizontal lines, T and A has 2 horizontal lines. So remember, these lines are presenting the bond between these two nucleotides. What are they presenting? The bond between these two nucleotides. Okay. If there is 2 lines, then there is a 2 bond between these two. If there is a 3, then there are 3 bonds. What type of bond? Let us see. It is usually the H bond. Secondary structure of nucleic acid was suggested by Watson and Crick. See, it is like a ladder type. They suggested double helical structure for DNA like this. 2 nucleic acid chains are wound about each other and held together by hydrogen bonds between pairs of bases. These two bases have hydrogen bonds. The two strands are complementary to each other. The two strands are complementary to each other because the hydrogen bonds are formed between specific pair of bases. A say T, G say C. A say T, G say C is connected. And how many number of H bonds are between A and T? Then you will say how many? 2. G to C. Then what you will say? It is 3. ATGC. Is that absolutely okay? ATGC is the number. No problem with this, right? This is very, very important. I'm trying to be very slow. I hope you are getting what I'm saying. ATGC. Okay. Moving further. Go slow. In secondary structure of RNA, single-stranded helix is present. Like here, 2 are present. Na? In RNA, no two, only one is present. Very, very important. DNA has a double helical structure. RNA is a single stranded helix. Single only will rotate is present, which sometimes fold back on itself. It folds on itself. RNA molecule are of three types: messenger RNA, ribosomal, and transfer RNA. Okay. DNA sends signal to first single strand of DNA. The two strands separate. Then the one strand replicate and form this mRNA. This entire process happens inside the cell's nucleus. Now this mRNA move out of nucleus, cytoplasm, and send message to ribosomal RNA. A ribosomal RNA means send message to transfer RNA, and then it is converted to protein. So protein synthesis का sequence क्या है? We start with DNA. DNA split like it was a double helical. It splits, separate. We have a cutting. We cut them into two. Single-stranded DNA. Now this single-stranded DNA transcripts its message to mRNA, messenger RNA, takes the message of that single strand. Go to cytoplasm and give that message to ribosomal RNA. Okay, ribosomal rRNA, and this rRNA then give message to tRNA, which is transfer. So now the protein synthesis starts from transfer RNA, tRNA to proteins are formed. Okay, so complementary DNA for following. This is first most question. If we are asked 5 dash and A, C, G, T, C, A like this. So as I said that A is bonded to T and G to C always. So if I am asked how many in a double-stranded DNA, how many A are there, so I will say equal to T. How many G are there, then you will say equal to C. This is going to be fixed. Is that clear? A, T, G, C. By this logic, the number of A's will be equal to number of T's and number of G's will be equal to number of C. And as you say, if there is C, it will be counter having G. T, A, T, like this. So if they give me a code that A, C, G, T, like this. From 5 dash they are writing. And they ask write a complementary DNA. So you just start opposite. From 5 dash you write 3 dash. Because as I said, no, complementary is that the double helical. A is bonded to T. So you replace A ke samne T. C, G, G, C, T, A. Here you use A, T, G, C. Okay. T, A, C, G, A, T, G, C, T, A, T, G, C, A, T, A, and then close it with 5 dash. Is that okay? Hana, it should be opposite. If given mRNA, Hana, made for transcription for following. So if they give you a DNA and they say, make mRNA out of it, then again you will do the same thing. 5 dash say 3 dash, 3 dash say 5 dash. That you will do. But instead of T, you will now simply replace U. A, T, no, A, U. Rest same, C, G. G, C. T, T is not right. T, A, okay. C, G, A, U, like that. Okay, and you will identify what it is. Now see this here, 5 dash say they have given it opposite. 5 dash A, C, U, A. This sequence is same. They just wrote it opposite. 3 dash pe aa raha hai. Yes, fine, single strand. Both options are correct. Is that exactly clear how to solve such questions? Okay, these are very important questions. You have to write the transcription, then a transfer this mRNA structure or the complementary DNA structure. Then you will use this. Now I see A is bonded to T, G is bonded to C. So I can say this is a rule, in fact, Chargaff's rule. A plus G upon T plus C is equals to 1. Okay, this is a famous rule. Rather, I should make it opposite. Chargaff's rule. Okay, what is Chargaff's rule? That this ratio is always fixed. From here, I can say that number of A units will be equal to number of thymine units in DNA. And number of G units will be equal to number of C units in DNA and RNA both. Number of A units will be equal to number of U units, right, in RNA. See, we replaced here na, instead of T we put U. So that is why instead of T I put U for RNA. This is a rule, right? As per the bonding, we found this rule. Moving further, and that's it. These are the type of questions that they can ask. The structures directly. What is an adenosine? This was asked. Among the following, which one is found in RNA? So, uracil structure was asked. Which of the following statement is not true? It has always double-stranded alpha helix. That is wrong. That is a line for DNA. It usually does not replicate. Yes. It is present in the nucleus of cell. It controls the synthesis of protein. Yes. Okay, they can ask the structures like this. Now, in mains 2022, the number of oxygen present in a nucleotide. Side is sugar plus base. Tide is phosphate plus sugar plus base. Sugar is connected to base at position 1 dash, connected to phosphate at position 5 dash, and then it is linked to another nucleotide through phosphodiester linkage from 5 dash and 3 dash, 5 dash, 3 dash positions. Number of oxygen present in a nucleotide form from a base, that is only present in RNA. So only present in RNA, which one? Uracil. Uracil. Okay. Total oxygens are asked in the nucleotide. So uracil has 2, and as it is RNA, so this oxygen has to be there. 1, 2, 3 here, 5, and phosphate has 4. 5 plus 4, 9. This type of question. Sugar moiety present in DNA RNA molecule is DNA deoxy, so beta D2 deoxyribose and beta D ribose in RNA respectively. Please solve this question. It is an important. Adenine constitute 0.16 mole fraction in a given single-stranded DNA. What is the mole fraction of uracil in the resultant RNA? First, if I talk simply for DNA, I will say A will be equal to T, G will be equal to C, like this, na? The only thing is in RNA, this will become U, rest will remain same. So A ka mole fraction is 0.16, so automatically T is 0.16, और U से जो replicate होगा, that will also have 0.16. So no need to solve. It has to be 0.16, 0.616. If I was asked for G, I will say 1 minus 0.16 into 2 upon 2 is the mole fraction of G equal to, same thing I can use, mole fraction of C, right? Mole fraction of C, cytosine. Do you all agree to this, that how it is solved? No problem. This is how you can use this rule particularly of G, like this. So what will be the value? This is 0.32. And 100 minus 32, 30, 70, 68. So 0.68 by 2. 0.68 by 2 will be 0.34. This is the mole fraction of G and C each. This if it was asked for G and C, but here they are asked only of uracil. For uracil will be answer 0.16. Is that clear? One more question of the same, a double helical DNA molecule. Yes, Sanjay, what is it? Tell.
Ma'am, can you please explain this question again, ma'am?
I just told you na A is bonded to T, G is bonded to C in DNA. So number of A units will be equal to number of T units. Similarly, number of A here will be equal to number of U here in RNA, because the only change that we have from DNA to RNA is change of T to U. Now they are already giving adenine is having 0.16 percentage. So automatically U will be 0.16 because they are equal. G will be equal to C. So if they would have asked G and C, right? So I will say that the rest is that and divided by 2 each will be G and C. Okay, Sanjay? So coming to one more question. A double helical DNA molecule is composed of 32 mole percentage of adenosine. The mole percentage of cytosine in this is, now see A you are given and C we are asked. A is given and C is asked. So again, A is equal to T. Solve, all of you solve by yourself. I'll wait for 30 seconds. Solution is there on the screen, but still you solve it by yourself. Let us check. Can you solve it? Basic thing is to be followed. And when you solve it, give me a thumbs up that yes, we are done. So simple questions will come. I have one thumbs up, but I don't know who is it. When you are done, give me a thumbs up. Okay, I have got 2. Very good. Okay. So you could do it now, na? This is how the questions. This is all about nucleic acid that could be asked. Very good. You are doing it, right? Okay. So that is all about nucleic acid. As far as vitamins are concerned, and yes, let me show the answer. I think answer was 18 percentage. That is the answer. Okay. Now let us go to vitamins. Let me start. The word vitamin is derived from vital amine. Vitamins are organic set of molecules required in very small quantity by the body for the proper functioning of our body. What could be asked? There are two types. One is the water soluble, other is the fat soluble. Water soluble vitamins are vitamin B and C. Vitamin B and C are water soluble, and vitamin A, D, E, and K are fat soluble vitamins. Okay. B and C are water soluble, A, D, E, K are fat soluble vitamins. This you need to remember. Water soluble, even if you take excess, na, like these days people eat lot of multivitamins. So if they eat excess, then also no problem, because extra body will dissolve in water and will throw it out in urine or feces. Okay. But fat soluble, if you take extra, there is a problem because they are not water soluble. So we cannot throw them out of the body. They are stored in our body in liver, okay, or in the fat tissues, adipose tissues. So, and their excess can cause again some diseases. So vitamin ADEK, okay, ADEK is fat soluble, or you can simply say vitamin B and C, BC are water soluble vitamins. Is that clear? You have to remember all these things. I have made the chart. NCRT has given the chart. Same thing. So vitamin A sources is fish, liver oil, carrot, spinach, dairy product like milk, paneer, butter. They are all vitamin A source. What are the diseases that will be caused in its deficiency? First is night blindness, also called ratandhi. Xerophthalmia, which is called the hardening of cornea of eye, such a painful disease. Vitamin B1, it is called thiamine. Okay, yeast, milk, green vegetable, and cereals. Those give vitamin B1. Beriberi in its deficiency, which has loss of appetite and retarded growth. Next is vitamin B2. It is called riboflavin, milk, egg white, liver, kidney. Now see, sources may everywhere there is milk. That is why our parents asked us to have a lot of milk when we were child. And if vitamin B2 is less, then the disease caused is called cheilosis. It is fissuring at corners of mouth. Like sometimes you may feel a dryness of lips and some cuts will appear and lips. Digestive disorders and burning sensation of the skin is due to deficiency of vitamin B2. Then is the vitamin B6. It is called pyridoxine. This is source is yeast, milk, egg yolk, cereals, etc. and grams. Now see, in its deficiency, we have convulsions, like I would say some loss of senses at time. Vitamin B12, which is meat, source is meat, fish, egg, and curd, right? So what is the disease caused in its deficiency? So we say pernicious anemia, which is deficiency of RBC in our hemoglobin. Okay. Vitamin C is called ascorbic acid. What is it called? Ascorbic acid. And this is obtained from citrus fruits like oranges, lemon, amla. Amla, what do you call in English? I'm not having some berry it is. And green leafy vegetables, they have vitamin C in them. Okay. And if we have vitamin C deficiency, then scurvy, bleeding of gums takes place. Then comes the vitamin D, which is exposure to sunlight. Of course, this sunlight should be the early morning sunlight, not like a 12 noon sunlight. 12 noon sunlight is full of ultraviolet radiations. It will form other skin issues. Exposure to sunlight, fish and egg yolk gives you vitamin D. Its deficiency causes rickets, which is bone deformities in children, osteomalacia, soft bones, and joint pain in adult. Yes, Sanjay?
Ma'am, can you please give the structure of ascorbic acid, ma'am?
It is very, very common. Ascorbic acid is this is ascorbic acid. Okay, sixth. So next. So these are the deficiencies and diseases. Vitamin E, vegetable oils like wheat, germ oil, sunflower oil, these all give you vitamin E. Increased fragility of RBC, that it starts breaking. Muscular weakness, vitamin E is an antioxidant. Like if you have a sufficient source of vitamin E in your body, aging will be delayed. Vitamin K, green leafy vegetables. It causes increased blood clotting time. So blood will clot late. And increased blood clotting time. That is what vitamin K does. Is that clear? Which of the following vitamin is helpful in delaying the blood clot? So it is vitamin K. This type of direct questions have appeared in the past. And that is it for this week and entire organics. So if you have any doubt or concern, please raise your hand. Soon we'll be uploading all these lectures on YouTube also. YouTube of Narayana group. So you can, once they are uploaded, you can have these lectures, those which you have left or whatever reason you were not informed on time, you can watch from there also. Okay. So that would be helpful to those who joined the program a little late. Hope that helps, right? So we have started the work on that and soon they will be uploaded. Yeah, please tell someone who has doubt, raise your hand, same process. So today it seems no one has any doubt. Is it? Okay, so I'm allowing you to unmute. Yes, Arman.
Ma'am, actually, 1 second, I literally forgot the doubt.
Okay.
1 minute. Has to do something at the starting, I believe.
Okay. If you are
After you extend the secondary structure, ma'am, can you just go back to
secondary structure of what?
the what's this? DNA and sorry, it's nucleic acid. Yeah,
Yeah.
this one. I think my
Yeah.
doubt was somewhere after this, ma'am.
Okay.
Yes, ma'am. This one only. The mRNA question.
Hmm.
Ma'am, how are these two structures forming, ma'am? A and B,
Oh,
right?
these. One is I have a pen. Na? Can you see the
Yes,
points
ma'am.
here?
I can see the point, ma'am.
And what if I write it like this?
Points change.
But the basic sequence internal remains same. Check A and B.
Yes, ma'am. I can notice it.
That is the trick.
Okay, so for RNA, this can be done, but not for
Yes.
DNA.
Uh, yeah, complementary, but usually if they will give this way, then it can be done. But depends. Matlab, just you have to be very detailed in this sequence should remain same.
Okay,
Usually
ma'am.
when they do this, na, they change the sequence.
Yeah, ma'am, got it.
If this same, then you can use it.
Hmm. Okay.
Okay. Anyone else? No one? Okay, Ibrahim, tell.
Ma'am, some additional vitamins are given in the material provided by our college. Are they not necessary?
I don't know which materials are there in your.
Like, no, like
But, huh?
the name is printed by Narayana and all that. So, about
Hmm.
B5, B7, and B9 is also given. Is that not necessary?
I'm talking to Ibrahim, right? Ibrahim,
Yes, ma'am.
I actually really I haven't seen the materials, so I have no idea what we are talking of. But as far as J Advanced is considered, I can assure you whatever will come will be as per NCRT. And if we go to NCRT, let us check what all are there. Okay, and that is what you have to compulsorily know these A, B1, B2, B6, B12, C, D, E, and K. Then not needed for J beta. I am not aware of MSETs. Okay,
Okay,
there I
ma'am.
don't know because just once you check your Telangana NCRT, like Telangana or Andhra NCRT you have
Mm.
now, in that you
Yes.
check the table. If they are giving those, then yes, for MSETs it could be required. For advanced, no.
Okay, ma'am. Thank you, ma'am.
Okay, thank you everyone. I hope this all helped you.